computational methods to analyze synthetic biology data. So first of all, I would like to point out that synthetic biology is highly dependent on the genetic information, and you need to process. You have to understand what are the genes and how they are expressed, and what kind of data you can be derived from this gene expression, why it is important, and what we can achieve by using all this information. So here's the kind of a representation of different components that you have to analyze genes and genomes. And for the standard gene, you have the structure as it is represented here. You have the enhancers, promoters, post-translational control, and microRNAs. Here, the standard gene has as a promoter the region where the transcription starts, and where the transcription factor binds, and a kind of untranslated region 5, prime 5, untranslated region prime 3, and kind of open reading frame where you have the encoded sequence for, if it's translated further, you have encoded sequence of the protein. So the goal of this is to have some kind of a system level understanding of genomes and gene regulation. So for the most part of that idea, we have to work on the understanding how the genes are expressed, and how we can modify this expression, and how we can inhibit this expression if it's needed, or what can be done in this direction in terms of the methodology. And here you have some information on genomes, like we have different genome maps. We have to align genes and genomes. We have to assemble different fragments in order to get the information on the genome. So it's more like sequencing. Then you have genes for encoding exons, introns, non-coding RNA, and RNA folding. So most of the part, if you are interested in a protein, you will probably look at the protein encoded exons, where the sequence of the protein is encoded. So modifying this or mutating this, you could also mutate the protein. And you also have a control regions, some promoters, enhancers, insulators, chromatin states, where you can try to understand in terms of the similarity, or how you can effectively use them, or the control regions are also important for kind of in epigenomics, when you get different modifications of the genes. And this all like some methylation profiles, ubiquitinylation profiles, and etc. Then you would have more information on the control system of the genome. Then you have also regulatory motifs, different regulatory motifs in the genes. You also have regulatory motifs in the proteins, different boxes. So those of you who is interested, what kind of regulatory motifs they have in a specific proteins, could go to the molecular biology textbook and you'll find all the information. There are a lot of different regulatory motifs, different systems. They have different regulatory motifs, different. The ATP ADP conversion motifs in the GPCR receptors. You have the motifs to activate specific cascades. You also have motifs which are important for protein folding. Some motifs are important for protein recognition, and etc. So there is a huge number of different motifs, and these motifs, ligand interacting parts of the protein ligand binding sites can be also catalytic motifs. So there are plenty of different motifs, like in HIV protease, there are some specifically triads, catalytic triads, which can also be considered as a catalytic motifs. These catalytic motifs sometimes important to recognize the protein traffic to go to be processed by the proteasome, or some motifs are important to interact with the nuclear pore and then transfer the protein to the nucleus, or some molecule to the nucleus. So depends on the what exactly you're searching for, and what exactly the mechanism of your protein by which they are utilizing their biological function. Also, the dynamics changing maps between cell types and across the development, some networks. So last time we already discussed what are the networks and how we can utilize them. And here you have a regulator, enhancer, and a target, ChIP-seq experimental protocol to generate these networks. Also, some motif mark combination predicting model. You can use some protocols like the sequence recognition protocols to search for these specific motifs in a protein or in a gene. So human, usually motifs, they are the parts of the protein, smaller parts of the protein. It's a kind of a signaling part which can be recognized by some molecule to ensure the biological function of this protein. And also human variations. You have also human diversity, populational genomics, and etc. Then we can also go further to the evolution, where we can analyze phylogenetics, phylogenomics, human ancestry, coalescence, etc. And some genome variation, like whole genome screening technique. Then we can use this to analyze the molecular phenotypes, organismal and molecular phenotypes. So here are the challenges in synthetic biology. What we have here, we have genome assembly. The genome assembly is you have some contigs here, and you will try to implement some algorithms to assemble the genomes of different organisms. Then you would have regulatory motif discovery, like shown here. You have different motifs in gene finding DNA, and then you will have sequence alignment, trying to figure out some similarities, some motif similarities. One can do also comparative genomics, compare data. Then we will have some database lookup, and then do the comparative genomic evolutionary theory. Evolutionary theory is mostly dealing with some kind of dendrograms, when you could cluster your sequences using some similarities indexes. So, and this would give you the idea how what is the evolutionary impact of for this particular protein of interest. You could also use the gene expression analysis to analyze the RNA transcripts, then cluster them using heat maps here, or Gibbs sampling, as shown here. Then you can do the protein network analysis and metabolic modeling, and finally you would provide like emerging network properties. So, all this can be integrated in chemoinformatics protocols as well. Then if you have the drug of interest, you would try to understand whether this drug is probably is inhibiting these specific proteins that might regulate the gene expression, or there are some mutagenic activities of the substance, and the drug would also create some mutations which are also harmful for this particular gene or genome, or the whole organ, or the whole body. Or if you have some mutations during the evolutions, how these mutations would affect the protein-ligand interaction for rational drug design and discovery. So you have many questions. Again, depends on the project. Sometimes RNA, especially microRNAs, they can be used as a therapeutic system. Then it is also important to investigate this mechanism, especially the gene silencing, and how this would contribute to the rational drug design. So the synthetic biology usually is the kind of experimental field, but highly reliant on the computational methods like computational systems biology, bioinformatics, chemoinformatics. So data mining, network construction here, protein network analysis, molecular docking as well. So some protocols also include the DNA sequencing to either modeling techniques to produce DNA sequencing, or the experimental techniques. There are some softwares like Genius or Plasmid Editor, where you could actually model the PCR reaction, trying to construct the kind of the recombinant DNA, and then you would sequence it and compare the sequencing results with the experimental data. But first, you could try to do this in silica. So this is just a main idea, and what is the main idea? Our central dogma in synthetic biology. The central dogma is in synthetic biology includes DNA, RNA, and the protein. You have the gene, and gene is transcribed. Then you have the RNA, and then you have translation mechanism, and then you will have a protein. Transcription, it will start also from the amplification, and then transcription comes along, and then finishes with the translation, and you will have the protein. So these phases can be modified, can be analyzed for mutations, for modifications, for the 3D structure as a protein, to understand how we could actually use this for which purposes. One could, as I already mentioned here, the 3D structure, and then you got the RNA, which will start the translation of this protein, fully folded protein. To expand the picture, sometimes it's a very schematic representation of the central dogma. You could expand the picture, and you will see that here you have also the cell. It goes within the cell in a chromosome, and the chromosome is a very complex structure. You have telomeres, you have different arms, p arms, q arms, and centromere in the center of the chromosome. And the chromosome is a very complex structure as well. It is made from the parts which are called nucleosomes. These are histones plus the DNA double helix molecule. And it is very heavily packed to produce some structure called solenoid. And finally, this solenoid is organized as a chromosome. Then this structure is representing the nucleus, where all the chromosomes are located, and where the replication of the DNA is taking place, and transcription of the DNA is taking place. So, what is the DNA, and what are the molecules which represents the DNA in more detail? Yeah. As you can see here, we have different bases of the DNA in the DNA, and they are complementary, bound to each other by hydrogen bonds. We got AT pairing and CG pairing, and these pairings have 2 or 3 hydrogen bonds, these hydrogen bonds hold 2 strands together. This allows a long opening and reclosing of 2, reopening and reclosing of these 2 strands. And you have anti-parallel strands from 5' to 3', and vice versa. Then the 5' is the carbon atom in a base here, and 3' atom is shown and also in a base. in a different direction. So it's oxygen atom here, and here carbon atom here, and you also have phosphate residues in a DNA structure. So this DNA molecule has also different conformers, like Z structure DNA, but we are not going into details what kind of conformers they have, but they are also very important to analyze. We have a collaboration with some universities in Moscow, and they are analyzing these conformers which are important for the mechanisms, for the pathology of cardiovascular diseases. So some different cardiovascular diseases are also associated with different structure, with different DNA structure. And we also have different bondings here, shown here, purine, pyrimidine here. So some are weak, some are strong here, like it's shown here. These are two bases when you have the exchange of thymine molecule to uracil molecule, then you'll have a RNA molecule. You convert the DNA to the RNA molecule. So what is the genetic information, and where it is located within the cell? So when you will look at the different cells, like eukaryotic cell and prokaryotic cell, the genetic material is located in different compartments of the cell. So here they're just located in the cytoplasm. You also have small DNA molecules which are called plasmids or episomes, which can be transferred from one bacteria to the other. And DNA is usually organized as a single chromosome, so you don't have a nucleus and mitosis. On the other hand, if you will take eukaryotic cells, the genetic information is usually organized within the nucleus in a special compartment, and the DNA is organized in multiple chromosomes inside the nucleus, and therefore you have a mitotic division. DNA also in a special DNA in eukaryotic cells is packed, heavily packed. And why you need the packaging of the DNA? Because DNA is very long, and the cell size is limited. So therefore you have to squeeze the space within the cell and make it more efficient. Then chromosomes are also produced because they're compressed, and chromosome, the single chromosome, is 50,000 times shorter than the extended DNA molecule. On the other hand, the DNA, using the DNA, before a piece of the DNA is used for anything, this compact structure must be opened locally, because when it's heavily packed, with histone protein is not really expressed. The genes cannot be expressed. It should be opened first, and then the genes, only then, after that, genes can be activated. So it specifies the role of accessibility, the state of the chromatin itself, and the role of different interactions with different transcription factors and different proteins which control this process. So here the packaging mechanism in more detail and what kind of proteins you have. This is the diverse epigenetic modifications. You can have here, for instance, epigenetic mechanism can be affected by different factors like the development in utero, in the childhood, or different environmental chemicals, drugs, and different pharmaceuticals. They are also very dangerous in terms of the impairment of epigenetic modifications. And the aging and the diet are also the factors which could produce different epigenetic profiles. So all this, as you can see, is important for the gene expression, and therefore, if one would try to understand different health endpoints like cancer, autoimmune diseases, mental disorders, diabetes, etc., you would try to relate all these points to the different epigenetic profiles. So here, when you will take the single chromosome, you might produce chromatin, and after stripping the DNA up from the histone molecules, you have the DNA which is methylated, and these methyl groups are epigenetic factors found in some dietary sources that can tag DNA and activate the response or repress genes. So different mechanism depends on where these methyl groups are usually added and in which part of the DNA, and this would either activate or repress specific genes. And histones are the proteins around which DNA can bind for compaction and gene regulation. So once the DNA is bound to the histones, the DNA is inaccessible and the gene is inactive. Then you would have some accessibility when you stretch the DNA up and the DNA becomes accessible and it activates the particular genes. And of course, different factors could. So all these factors are important for the gene expression, and if you will try to analyze these epigenetic factors, you could have also the additional mechanism to control genes. So the other, the more information on the diverse epigenetic modifications are that more than 100 different histone modifications are present. They can include some histone proteins like H3, 4, 2A, and 2B. There are some amino acid residues which are affected, in particular lysine, lysine 4 as a K4, K36. Chemical modifications are present, like methylations, phosphorylations, ubiquitinations. These modifications are also present in the protein as a post-translation modification. Some numbers, different methylated groups can be added like this. In addition, there are some methyl CpG, like CpG islands, are very important for to control the gene expression. They are called the CG sites, and these are the regions of DNA where cytosine nucleotide followed by guanine nucleotide in a linear sequence. And these sites are frequently found in the genomic regions, and they are called CpG islands. So cytosine in these islands can be methylated to form 5-methyl cytosines. And in mammals, 70 to 80% CpG islands are methylated. And this methylation can change its gene expression, and the mechanism is a part of the larger field studying the gene regulation and to understand the epigenetic profiles in for different pathologies, for example. Some methylated cytosines are often mutated to thymines. So in humans, 70% of different promoters located near the transcription start site of a gene containing CpG islands. So these CpG islands are very important to understand the mechanism of gene expression and at which particular conditions they are methylated or they are impaired, and therefore the epigenetic profiles are different. There is a software which can give you the information or the analysis of CpG sites. One could analyze the distribution of CpG sites and cytosine mutations in mammals. In mammal genomes, these sites are typically contain 300 to 3,000 base pairs in length, so they're quite big, and approximately 40% of promoters are found near these CpG sites. Also, CpG sites are important to housekeeping genes in vertebrates. The housekeeping genes are the genes typically required to maintain the basic cellular functions of the organisms and pathophysiological, under normal and pathophysiological conditions. There are different housekeeping genes. The common housekeeping genes are different transcription factors like ATF transcription factors, BTF transcription repressors, RNA splicing proteins, BAT1 protein, transcription factors like EF1 transcription factors. So most of them, they are translation factors, transcription factors, repressors, and RNA splicing proteins. They are, of course, very important to maintain the genetic stability. One could also see some proteins like ribonuclear proteins, A2B1, as a possible protein of housekeeping, as a housekeeping protein to maintain the genetic stability. So this methylation profiles was found in some pathologies like cancer, and especially when there is a hypomethylation, hyper and hypomethylation profiles are detected. For example, in colorectal cancer, there are usually 3 to 6 driver mutations and 33 to 66 hitchhiker or passenger mutations. And they are all associated, might be associated with the CPG islands. And also some studies are shown that more than 140 genes are associated with colon cancer, and they have hypermethylated promoters, so which means that they have different expression levels in comparison to the normal cells. In our case, when we analyze the proteins, usually the methylation profile is given the proteins that sometimes a decrease in stability, which was assessed by the free energy using the Monte Carlo algorithms. Also, some DNA repair genes are also associated either with hypo or hypermethylated promoters in cancer. Such as REP-P1, FEN1 promoters were hypermethylated, and they were overexpressed in numerous cancers. So the pattern is when you have a hypermethylations of the genes, this might contribute to the overexpression of this particular genes in the numerous cancers, such as leukemias, neuroblastoma, testicular and other germ cell tumors, Ewing sarcoma, prostate and stomach cancer, and lung cancer. So as you can see, the methylation profiling is very important to understand the gene expression in various cancer. And then one could also upscale this profilings to what are these particular proteins that are genes that are overexpressed, and what are the proteins that might be probably mutated in a cancer, and one could find some correlation between these processes. The methylation of these sites is also known to to increase with age, and there are some publications that point out that there is a strong effect on DNA methylation level on tens of thousands of CpG sites that can define the kind of biological clock in humans and apes. There are also some unmethylated CpG sites that can be detected in toll-like receptors like TLR9 on plasmacytoid dendritic cells, natural killers, B cells, etc. And the toll-like receptors play a fundamental role in pathogen recognition, in activating of innate immunity. So very important mechanism in innate immunity and pathogen recognition. There is also some information that the methylation profiles in CpG islands are associated with memory, and there are a lot of publications that the memory decline in some patients with psychiatric diseases. They have shown that you have differences in epigenetic profiles due to different gene expressions. Also, there are some studies to investigate the link between the methylation of CpG sites and the increase of reactive oxygen species activity. Some studies point out some proteins related to this mechanism, like ApoBec proteins, proteasomal machinery proteins, etc. So sometimes there is also CpG loss, and it was observed in response to some some stimuli like radiation. So all these mechanisms are very important to investigate this epigenetic variants found in different conditions. One could also analyze nucleosome positioning, where these nucleosomes are located within the cell. This is relevant for some types of cancer and so on. So, what is a connection between SNPs and how we could actually use this information for synthetic biology? What are the methods we could use to analyze different mutations which are found in genetic material? So the SNPs are the single nucleotide polymorphisms, which means that you have the substitution of nucleotide as shown here. So one substitution would give you some variations in in the genes. So gene is expressed, and if this substitution is missense, non-synonymous, then you would come up with a substitution in an amino acid, with specific amino acid in a protein. Therefore, you can classify the types of different SNPs. You have SNPs of non-coding region, which is almost have no effect on the expression. Then you have SNPs in a coding region, and these are subdivided into two, like synonymous, when you get the same codon, the codon which is affected, but it stays the same for the same amino acid, so the protein structure is not affected. And you get non-synonymous SNPs, where you have the substitution which will change also the amino acid in a polypeptide chain of the protein. And this is also called like missense mutation. So missense mutation, what we are looking for, because we are changing the structure of the protein, like shown here. If you have chromosome, you have the gene coding region, then you have the substitution of a common genotype, and then you have a protein. If it's just a common genotype, you have no difference. If you have variant genotype like this, you have a substitution, the SNP site, then you would have a altered protein in a different part of the protein. If it's very important for binding, for instance, as a protein ligand binding site, so this would become affected, and you would have differences in the protein ligand interaction. So SNPs can help to explain differences in a susceptibility of different range of diseases, increasing some to understand what risk is related, for instance, for macular degeneration and stuff like that, in the CFH gene, which is related to this process. Some SNPs were found in APOE genes, which are also related to some associated risks for the development of Alzheimer's disease and age of onset of this disease. So, as you can see, some particular pathologies are related to specific SNPs, and it is also very important to find some genes that might interfere with the protein function. Here is also kind of a scheme where you see like different genotypes, human genotypes. Then you would have a mutation as a SNP. Then it would produce the SPG methylation and the methylation in the SPG islands, and this would affect the transcription factor for the particular gene, and the binding site is affected for this particular transcription factor, and this in turn would also affect the expression of this particular gene. The expression is impaired, might be impaired. So this would be very important to analyze this kind of processes, kind of populational genetics, if you upscale it to the population, or in a small cohort of people. So here we are trying to combine the mutations and the epigenetic profiles to understand how this would affect the expression of the particular gene. Sometimes these SNPs with an allele frequency less than 1% are called single nucleotides variants. And these variants are important as they are rare mutations in some germline and somatic cells. So the frequency of the SNPs, usually more than 600 million SNPs across the human genome, was already detected in the world's population. And the typical genome affected differs, and it differs from the reference human genome at 4 to 5 million sites, most of which consist of different SNPs. So actually, these SNPs, the lecture, I have a lecture. And therefore, the SNPs are affected to, important to link them to the particular phenotype found in different populations. So how we can actually generate the SNPs, or why it is also the concern of synthetic biology. And here I have the information about the polymerase chain reaction, what it is for, and how we can use in the synthetic biology. Polymerase reaction is very commonly used methodology in synthetic biology, especially when you want to amplify the part of the target DNA, as shown here. We have some region, then we would like to amplify. So for this, we have to use the PCR reaction. And the PCR reaction is a kind of very well optimized in our laboratory. We have for this you have to know what kind of template you have to amplify, and probably generate some novel aptameric molecules, or you can introduce some mutations to the DNA and trying to understand, at least to model these mutations in terms of the stability and the interaction with some transcription factors. So as I already mentioned here, one could also simulate this process, at least using the PCR reaction, when you have the small part of the DNA as a DNA template, and you could modify this template by introducing some mutation within this template. If you will design the primers, then you could design them in a way that you would have SNPs within this particular primer, they would just, this is the primer, this is a forward primer, this is the reverse primer, and you have also the nucleotide as a building blocks to perform the amplification. To perform the amplification process. Then you would use the original DNA to be replicated in some nucleotides. Then the first part, the first stage is the denaturation, when you will program the chamber to 94 from 94 to 98 degrees for at least 20-30 seconds. Then you will have your DNA will break down, and you will try to, in the second stage, you would try to decrease the temperature, and the annealing is taking place. The annealing means that your primers would bind complementary to these specific areas of your DNA. And if the primer is mutated, that will also produce the mutated part of the region. So then amplification is elongation is taking place, extension and elongation at this specific temperature, using some Taq polymerase, Thermophilus aquaticus polymerase, which is thermally stable, so you could actually amplify a specific site. And then you would simply program the number of repeats, and after some time you would finally hold the amplification, the whole procedure, hold the reaction to from 4 to 15 degrees for the indefinite time, for the short-term usage of the PCR products. So this is a kind of universal, very simplistic scheme of the PCR process. Those of you who is interested, I would suggest to take part in very fascinating projects we have on gene amplification and how these genes are probably affected by mutations, by different mutations. We also have a lot of data coming from oncologists. Together with Professor Kravtsov, we already optimized the chromatopsia model of the cancer, where you could simply focus on some genes and how these genes mutated, or probably you will mutate some genes, or if there is a specific project concerning the aptamer generation, that one could also generate the aptamers using the PCR. The only thing is you could use a lot of different primers to design the various aptamer molecules, and then produce some kind of a select, which will give you the leverage on the specific aptamers that will bind to a specific molecules. In our case, this is nivalenol, which was found found in in some pathogenic fungi as a toxic agent. So actually, this is the PCR reaction. We could also try to investigate the stability of different fragments. For instance, if we have different mutated regions within the DNA molecule, we will analyze the stability using thermogravimetric analysis, or for instance, various very simple like incubation, different incubation techniques at different temperatures, at different pH, to see whether we will produce different fragmentations, and using some physical chemistry technique like spectroscopy to analyze these fragments. The fragmentation rates can be also achieved by different simulation profile. The stability of the DNA fragments can be also analyzed in silica, in the molecular dynamic settings, when we could simply run different molecules at different temperature, different pressure, different salt concentration, and so on. So we can use different solvents to investigate how these molecules would, how these mutations would affect the stability of this specific region of the DNA, and how all this would interfere, at least at the stability level, would interfere at least with the expression data. Because the hypothesis would be, if you have more unstable fragment, then probably this would decrease also the expression level of the gene, and at the same time it would also increase the mutation rate of this segment. So would be a very interesting project. So a lot of possibilities, as you can see, even by using the PCR reaction to analyze different DNA molecules. One could also achieve some kind of hybrid generations if you have different primers or different nucleotides which are linked to a specific molecules. Then, for instance, nanotubes or some reporter molecules, then you could generate some hybrids. Or you could probably integrate some unconventional nucleotides that might be also integrated into the system. So at this point, you could generate different hybrid molecules to see the binding, the stability, the interaction, either with the folding, how it would also create the molecular folding of the DNA, and etc. So the application are endless. Okay, and here I would like you to suggest. I would suggest you to have a look at this very interesting software, which is called APE, a plasmid editor. It's free of charge. It's the open source. It's now widely used in synthetic biology to run the conventional PCR reaction, as shown here, when you have when you have a plasmid, usually this plasmid is highly used for the gene amplification and modifications. You could introduce different parts of the DNA into the gene of interest. You can modify, you can substitute the promoters, the open reading frames. You can add different selection markers. You can also add the reporter molecule, like you have here, like GFP, the reporter molecule. You can substitute for different reporter molecule, etc. So usually we have you can do also subcloning, and you can also run the PCR. When you have this template, you could amplify the small part of this vector and see how it is actually visualized and at on the agarose gel. You get the ladder, and by size, you can see which fragment you can produce by running in silico PCR. The program also has a database for the restriction sites, the restriction endonucleases. So you have to make sure that the restriction sites are you are using the right restriction sites to exchange for your region of interest, and then after the successful PCR, experimental PCR, you could purify and sequence your genetic variant and the substitution that you want to introduce to your cloning vector. So this software is also possible to use to investigate the PCR reaction. And here you could also program this, optimize the in silico the PCR reaction by changing the melting temperature, decrease the number of dimers, and so on. So some other parameters also can be analyzed, and after all of this optimization, there is a high chance that the PCR will work. There is the other software which is called Genius. It has a lot of inbuilt parameters like blast, so you can do the blasting by using the NCBI databases to find similar fragments. You can also express the genes and translate the fragment to create the 3D structure of the protein, and then you probably see how this mutation would actually affect the gene mutation, how it would actually affect the protein 3D structure and the function. So it's also very powerful. You could also use this software for your research project in synthetic biology. So in a template, the DNTPs, desoxydenucleotide with instead of DNTPs here. And these DNTPs, they are marked with the different reporter molecule. So they are modified in order to terminate the ligand in order to terminate the PCR process, a particular nucleotide. So DDNTPs are able to chain terminate, instead of, if you perform the PCR reaction, you just add DNTPs and the reaction will not terminate. So this is the idea. You have a template, you have a primer, and then you will start to amplify the part of your interest here. After some time, you would, once it's the nucleotide is bound to your template, it will stop, and you will have different fragments which are giving you different signals on chromatogram. You have laser, you have detector, and the capillary gel. So this mixture will go through this capillary gel, and you have the also the different fractions of your product, PCR product. Then you will analyze it with the laser and the detector, and laser detection of your laser detection will give you the information of your nucleotides representing this particular sequence. So quite effective for the small, relatively small fragments. This technique was developed by Frank Sanger. That's why it's called Sanger chain termination method, and widely used to sequence different DNA products. So the sequence reads usually 500 nucleotides, and the Sanger sequencing is still widely used in public health departments to sequence this small DNA, especially in last years when it was needed to sequence the spike protein from SARS-CoV-19 for the surveillance of this outbreak. Yeah, and this technique is relatively slow and cannot give you the ability to process the whole genome. Only the small part of this PCR can be processed. It also include poor quality, relatively could include relatively poor quality of the first 15-40 bases of the sequence due to primer binding, and deteriorating the quality of sequences after 700, 900 bases. So actually, the primer will give you the poor quality, and the quality will also deteriorate after 700 to 900 bases. So more than 900 is not really feasible to sequence because of the very poor quality. Current techniques could directly sequence relatively short DNA from 300 to 1000 nucleotide long DNA fragments in a single reaction. There are some modifications of Sanger sequencing. They're called fluidic Sanger sequencing. Is a lab on a chip applications, and for the relatively small sequencing DNA, it's also possible to analyze. One useful application is to use this technique to determine specific single nucleotide polymorphism, single strand conformation polymorphism, heteroduplex analysis, and short tandem repeats analysis. So actually, there is a lot of information one could obtain from this technique. Resolving DNA fragments according to the differences in size and conformation is also a critical step in studying these features of the genome. This is a standard procedure, and the next would be to use the NGS, next generation sequencing analysis, which is this is also called massive parallel sequencing of this DNA to use massive parallel processing. And this was the name to discriminate between the chain termination sequencing, because the next generation is a kind of a technique was developed later on, and it has the possibility to sequence sequencing of 1 million to 43 million short reads containing from 50 to 400 base pairs in each per instrument. So on one hand, next generation sequencing is extremely useful to sequence multiple genomes and big parts of the genome. On the other hand, like exomes, but on the other hand, the reads are smaller than for the chain termination sequence, where we have the sequencing of relatively bigger fragments of the gene, like up to at least 1 kb. Many NGS platforms differ from in engineering configuration. Right now we have various platforms like Illumina platform is kind of prevalent. Roche 454. Then you have the new generation platforms, which are called Oxford Nanopore, next generation sequencing platform. Different techniques are also introduced using pyrosequencing, sequencing by synthesis, sequences by reversible terminator chemistry, sequencing by ligation, mediated by ligase enzymes, and so on. So in here, it's a kind of NGS technology starting early in 1981. This was the first trial to use the DNA sequencer. Then Affymetrix platform was introduced, like this one, the chip based platform in 1991. first microarray publication was done on Arabidopsis italiana, and the rise of the gene bank database has spurred the research in these directions. At the time, there were some companies building DNA analyzer for the Human Genome Project, and DNA sequencing became industrial. So in that time, this was the first genome which was sequenced completely. The human genome was sequenced completely, and the researchers created some kind of a HapMap project to analyze the sequenced genome. And first gene expression arrays were introduced by Illumina and published in Nature. So now we have some new generations of Illumina platforms. Even nanopore DNA sequencing is also widely used so far, and we have projects where we have sequencing using this methodology, and this also it significantly is reducing the cost and effectiveness of the sequencing. So just to give you the update on the platforms, what kind of sequencing platforms we have, and this is just one of the platforms, the Affymetrix microarray, where we have the total RNA with the poly A tag for the stability. Then we will use the reverse transcription if you want to analyze the transcriptome. We use the reverse transcription, produce the complementary DNA, then in vitro transcription would produce bioattenuated complementary RNA, which is then fragmented, and in this part would transfer hybridized to the gene chip expression array. So the hybridization takes place, and after washing and staining, you would have a biotin scan, biotin reaction, biotin-avidin reaction with some reporting molecules that can be detected, and you would have something like this, this plate, like this chip with signals, which this signal can be analyzed and related to the hybridization of this particular fragments. So actually you are trying to match the transcriptome to via the hybridization to different genes which are sitting on the chip as a target molecule. So this is a kind of a biotin activated assay, biotin used assay, and after the analysis you would create some kind of networks or the information about the expression, the gene expression, because the transcription is actually the level of the gene expression in the cell. So this gen chip is a kind of old technique. Now you, as I already mentioned, you have this nanopore, and in nanopore you have a pore where the molecule could pass through, and by analyzing the current of this molecule, one could determine also some sort of peaks which represent different nucleotides in the polynucleotide chain. After that, one could determine the right information about the nucleotides in this polypeptide chain. So these are the projects we have concerning the SNPs. I think this would be better if we can discuss them in the next lecture. I have some examples, what kind of SNPs we can analyze. And either we are using the protein molecules here, or we are also using the genes where we can introduce some mutations in the genes according to the mutation rate known from the literature, and we will try to run some sort of in silico PCR reaction to determine what kind of protein is affected by the mutated gene. So then we can just discuss it in the next lecture. If you have any questions, I would be happy to answer.